Hello, my name is Carol Leibel, and today we're going to examine cell communication. Cell communication is important for both multicellular organisms and unicellular organisms. Cell communication helps to coordinate the activities and events necessary for multicellular organisms to develop from a zygote into trillions of cells, to differentiate those cells, and to coordinate those cells. In unicellular organisms, cell signaling is important in finding different mating types for sexual reproduction when the conditions are right. They have also found prokaryotic cells, in certain instances, can undergo cell communication. That being the case, cell communication is a process that has been conserved throughout the course of evolution and is an important process. Let's look at communication between unicellular organisms. In this case here, yeast cells. We have found that in yeast cells there are two different mating types. There's type A, and mating type A has a receptor site for a factor that's produced by the alpha mating type. Now, mating type A also produces its own little mating factor, and it's called the A factor. Now, the alpha mating type has a receptor site for the A factor produced by the A mating type. And so when both cells are near one another and they're producing different mating types and they're received, what's going to happen next is that these cells are going to move towards one another. And when they do, they're going to fuse and become a diploid cell. Now, once they become a diploid cell, they're going to undergo meiosis and produce 4 haploid cells. Each haploid cell is going to be genetically different from the parent cells, and so this is a source of genetic variation. Now, cell communication can happen at different levels. For example, it can happen at a local level. An example of this is called paracrine signaling, in which we have a cell that produces some sort of factor, and then those factors are released. The factor may cause neighboring cells to undergo mitosis and to grow, or it may cause those cells to actually to differentiate and become certain kinds of specialized cells. We also can find localized communication happening with neurons. So in the little diagram that we're looking at, we have a neuron, and it is going to activate the second neuron. Now, in order for that communication to take place, the first neuron is going to produce molecules called a neurotransmitter, and that little space between the two neurons is called a synapse, and those neurotransmitters are going to diffuse across that synapse attached to receptor sites. Once that attachment takes place, this second neuron is going to be activated. Again, this is an example of localized communication, two cells very close to one another. That communication can also happen through direct contact. So here we have two animal cells, and you'll notice that there are pores between the two cells. Those pores are called gap junctions, and it allows for molecules to move from one cell to the other cell in communication. Now remember that plant cells have that big old cell wall, and so it's not called a gap junction when you talk about plant cells, but rather it's called a plasmodesmata. But the function is the same. It allows for communication. We also find that communication can happen between a macrophage and a T cell. We have direct contact, we have a receptor site, and we have a little protein out here on the cell surface. And once this attaches, this macrophage can secrete something called interleukin, which will cause this T cell to do mitosis, mitosis, mitosis in part of the immune response. So communication can happen on a localized level through direct contact as well. There can be communication, though, via a very long distance, and this may involve the use of hormones. Animal cells will often release hormones directly into the circulatory system. Those hormones, once in the circulatory system, will travel to a target cell. That target cell will usually have little receptor sites, and some sort of response will take place. Now, plant cells don't have a circulatory system like many animals do. So quite often those hormones are going to be packaged up into little vesicles, or they actually release hormones into the air to affect other parts of the plant, like ethylene. So let's look at different types of animal hormones. There are uh, 3 different types. The first type is our little steroid hormones. As you recall, steroids are made out of 4 interlocking rings. And what causes one hormone to be different from another are these little functional groups attached to the 4 rings. Some examples of hormones you may be familiar with is testosterone and estrogen. The one thing about steroid hormones is that they are lipid soluble. They have the ability to go directly into the cell and exert their effects. Other types of hormones do not have that ability to go directly into the cell. For example, the hormone thyroxine produced by your thyroid gland is a modified amino acid. That modified amino acid turns out to be tyrosine. And so it's a pretty small little hormone. On the other hand, you can have a very complicated hormone like the protein insulin. And insulin, like thyroxine, cannot enter the cell. And so it exerts its effect by interacting with receptor sites. So this is a little video that shows you the action or how steroid hormones work.
A steroid hormone, such as estrogen or testosterone, acts by entering a target cell and turning genes on or off. Steroids usually act rather slowly because it takes time for gene products to build up or become depleted. The steroid hormone passes through the plasma membrane and binds to a receptor protein in the cytoplasm. The hormone receptor complex enters the nucleus and attaches to specific regulatory sites on the target cell's DNA. There it acts as a transcription factor, a gene activator. A messenger RNA molecule is transcribed, which is then translated into a protein, an enzyme, for example.
All right, so let's look at or examine the action of non-steroid hormones.
Non-steroid hormones, hormones derived from amino acids, oxytocin and epinephrine, for example, work via signal transduction pathways to activate or inhibit proteins within cells. These hormones can trigger an immediate response in target cells by turning cell components on or off. A non-steroid hormone binds to a receptor protein in the plasma membrane. The receptor activates a signal transduction pathway in the cell. A series of relay molecules transmits the signal to a protein that carries out the cell's response. In this case, an enzyme is activated. A different non-steroid hormone might trigger a different response, perhaps turning on specific genes.
So there are 3 main stages of cell signaling. The first thing is called the reception, in which the signal molecule interacts with the receptor site. Once the reception takes place, then there is a biochemical pathway that is activated, and a series of chemical reactions take place. That series of chemical reactions is called the transduction. And then finally, there is some sort of cellular response that occurs. Okay, so let's begin with the reception. The reception begins with the signal molecule. Remember, it's going to be a non-steroid molecule interacting with the receptor site located on the outside surface of the plasma membrane. The signal molecule never goes inside the cell itself. And if there's no receptor site on that plasma membrane, that signal molecule will ignore that cell and never interact with that cell. So that receptor is extremely important. When it does interact with the receptor is going to undergo some sort of physical change. And that physical change then will cause some sort of interaction with cellular components. Receptor sites are usually proteins, and again, they're found inserted into the plasma membrane. So let's look at this little video on the reception.
Cells communicate with one another by means of chemical messengers. The cell targeted by a particular messenger has a receptor protein that fits the shape of the messenger molecule. The binding of the messenger to the receptor often triggers a chain reaction involving other molecules, a signal transduction pathway, that relays the message from one molecule to another inside the cell. The activated molecules at the end of the pathway perform a specific activity inside the cell, such as catalyzing a certain reaction, rearranging the cytoskeleton, or activating a specific gene.
So we're going to look at three major types of membrane receptors. The first one is called a G protein linked receptor, and it's called that because it's going to interact with something called a G protein and link up with it. So here's someone's rendition of what it looks like. And up here, that's going to be the receptor site for the signal. And then down here, this is where it's going to interact with that G protein. And then we have the tyrosine kinase receptors. You'll notice that this molecule actually is composed of two parts, two monomers. What's going to happen is that they are going to come together, and they have these little amino acids sticking out here called tyrosine, and they're going to phosphorylate other molecules in the cell. So therefore, it's called a kinase. And then last but not least, we have the ligand-gated ion channels. They're going to open up via a little molecule called a ligand. So let's look at the G protein linked receptors. They can vary in size, but quite often they are made out of 7 little polypeptide helices. There's going to be a receptor site for a particular signal, and then here is where it interacts with that G protein. They are found in the plasma membrane. So here is a pathway taking place. There's the signal molecule. It has to fit precisely. So it fits with the G protein linked receptor. Notice how it's going to change shape. And when it does, it's going to interact with a G protein. The G protein right now is inactive, but once it interacts with that receptor, it's going to become active. And you'll notice that here comes a molecule called guanine triphosphate, which has guanine instead of adenine, and it will activate the G protein. The G protein will interact with a particular enzyme to activate a particular biochemical pathway. Once that happens, the G protein is released, and a phosphate is released, and we start that all over again. So let's now look at the tyrosine kinase receptor. You'll notice again there are 2 monomers, and each one has 3 little amino acids sticking out called tyrosine. They will pick up a phosphate from ATP, and now they're going to transfer that phosphate to some proteins. The one difference about this particular system is that it can activate more than one little protein. So it can activate a number of biochemical pathways simultaneously. So let's look here. We have let's look at this little diagram, and you can see here are the signal molecules. Here are the proteins that are getting activated by being phosphorylated, and those phosphates again are coming from ATP, and some sort of cellular response will take place. So here's a little animation showing you how that takes place. There's the signal molecules attaching to the monomers. It slides together to become a dimer. The little tyrosines are phosphorylated from ATP, and this is all happening in the plasma membrane. Now it's going to transfer the phosphates to different kinds of proteins and activate a number of different biochemical pathways. Kind of cool, I think. And notice how that signal molecule has to be destroyed, or it would just continuously activate that particular receptor. All right, so let's look at the ligand-gated channels. So here is this little gate, and this little gate can be opened or closed. But in order to open the gate, there is a small little signaling molecule that is needed. And when it attaches, it causes the gate to change shape and become open. And usually it will allow then a flood of ions to cross the membrane. Those ions quite often are calcium. What is unique about this little system is that these little gates can be found in the plasma membrane, or they can be found in the membrane of organelles like the Golgi apparatus or the endoplasmic reticulum. So here's a little animation showing you the ligand attaching and the little ions just flowing across. Now, what they have found in examining these hormones is that quite often there are a couple molecules that appear over and over again. And one of these molecules turns out to be something called cyclic AMP. And because that particular molecule appears over and over again, they call it a second messenger. Two other examples of second messengers include calcium ions and something called inositol triphosphate. Now, in order for cyclic AMP to be synthesized, it requires an enzyme called adenylyl cyclase or adenylate cyclase, depending upon which textbook you're looking at. And this is what happens. First off, you have ATP, and adenyl cyclase is going to remove two phosphate groups, and then in doing so, it forms this little ring structure outside the pentose sugar, and this is what cyclic AMP looks like. However, if you break that little ring by adding some water to it, it can just become AMP. So it's very easy to convert ATP into cyclic AMP. So here is an example of how it works. Here's the G protein linked receptor attaching with the signal molecule. Notice it's going to change shape, and in doing so, it's going to activate that G protein. Look how GTP is going to be involved. The G protein is going to activate that enzyme. Another name for it is called adenylate cyclase, depending upon the book, either adenyl cyclase or adenylate cyclase. The ATP is going to be converted into cyclic AMP, removing two phosphate groups. And now once the cyclic AMP is formed, it's going to activate the biochemical pathway. Now here's an example of calcium and inositol IP3 being used as a second messenger. On one side, you can see that IP3 is being made, and it can involve either the G-linked protein or tyrosine kinase receptors. And this is kind of interesting because they're showing you that the inositol triphosphate is going to interact with that gated channel, open the channel, and allow calcium to come flooding out, which is going to activate another little messenger called calmodulin, which will activate a particular biochemical pathway. What you would need to know on the AP exam is that what would happen if something goes wrong, or what flows from one to the other. You do not have to have this memorized at all, but you have to understand what it means to be a second messenger. All right, so let's talk about transduction. So reception has occurred, and so the next thing that's going to happen is that the signal is going to be, a biochemical pathway is going to be activated. Now, quite often, it's going to involve a number of kinases, which means that it involves the movement of phosphates. And if we're looking at this, we can see, well, it activates kinase 1, kinase 2, kinase 3. And you may be asking yourself, why don't you just stop with that one right there? Why do you have to go through so many different kinases when all you're really doing is moving phosphate? But every time this occurs, it will activate the next step by a factor of 10. So it's like a cascading effect. And so therefore, when you get down here, instead of just activating one kinase, we have, instead of just activating one little kinase, this would be activate 10, this would activate 100, and so forth. So what you're doing is you're activating a lot of these kinases all at once, so the response can happen quite quickly. And so it's an amplification reaction. Once the kinase is activated, then finally what's going to happen is that there's going to be some sort of response. That response may involve the activation of a transcription factor. It may involve the activation of an enzyme, but something is going to happen as a result of these kinases being activated. So here's an example of transduction.
Cell-to-cell signaling, with molecules carrying signals from sending cells to receiving or target cells, is a key mechanism in the coordination of cellular activities. In most cases, a signal molecule acts by binding to a receptor protein in the plasma membrane of the target cell and activating a series of changes, a signal transduction pathway, that triggers a specific response within the cell. A signal molecule binds to a specific receptor in the plasma membrane of the target cell. This activates a series of relay proteins, which in turn activates a transcription factor. The activated transcription factor attaches to the DNA and triggers transcription of a specific gene. The mRNA message is translated into a new protein, which changes the structure or function of the receiving cell.
So now let's look at what kind of responses we could have. We could, as we mentioned, activate a particular enzyme, or we could synthesize a particular enzyme or protein by activating a particular gene. When cyclic AMP, calcium, or inositol triphosphate is activated, it's generally followed by interacting with specific enzymes to start a cascading effect of a particular biochemical pathway to amplify the product. So let's look at different kinds of cellular responses. So you have your signal molecule, you have your transduction pathway, and you have some sort of response. But if you look at this cell next to it, you can see that the response is different. And the reason the response is different depends upon what molecules are found in the cell. It is indeed possible that it could actually activate two different biochemical pathways. There could indeed be a hormone that instead of activating a particular biochemical pathway, it produces an inhibitor that will actually shut down a biochemical pathway. But the point being is that, first off, you have to have certain receptor sites. And then inside the cell, what molecules you have inside the cell will be responsible for what kind of response you'll end up with. So here's an example of a signaling pathway in a plant. This is a most interesting sort of signaling because the signal is light. When light strikes a particular receptor called a phytochrome, that phytochrome changes shape. And once it changes shape, it causes the production of something related to AMP, but it's GMP. And then that causes the activation of a kinase, which activates a particular transcription factor, which now causes a gene to be produced. Or not produced, but transcribed. Over here, calcium comes into this, acts as a ligand, opens up the channel, calcium comes in. Again, the calcium activates a particular kinase. The kinase activates a different transcription factor. A different gene is going to be transcribed. And now these products that are being transcribed are going to cause the plant to become green. Sounds a little funny, but hey, plants do indeed use cell signaling much like animals. So a scaffolding protein are large relay proteins in which other proteins like protein kinases are attached. Scaffolding proteins are also now found a part of cell communication, and because they can hold more than one protein, they increase the efficiency by grouping different proteins involved in the same biochemical pathway to be activated. All right, so we want to look at one more time the role of intracellular receptors, and that is the use of steroid hormones that go directly into the cytoplasm, attached to a receptor protein. So there's no receptor here on the plasma membrane. Usually that receptor protein will now go into the nucleus, and it will turn on or activate a particular gene that will be transcribed, and a particular protein product will be made. So that's how steroid hormones work. So now we have a nice little video that kind of, if you will, it summarizes all the things we have been talking about.
Signals from our environment are most often first detected by the brain. When someone is frightened, a stress response is initiated by their reaction to an environmental signal. But many signals don't stop here. A signal received by the brain can be passed on to many types of cells throughout the body and have widespread effects. Signals get sent around the body by traveling through the nervous system or bloodstream, going from cell to cell. You could think of the cell's sole purpose in life as keeping signals going or initiating new ones in response to the environment. Once a signaling molecule from the bloodstream reaches a cell and docks, the signal is passed into the cell. Like a miniature mousetrap game, the cell is filled with complex contraptions called proteins that bump and nudge each other to keep the signal going until it reaches its target. In this case, it's the production of yet another signaling molecule that will get released into the bloodstream, dock on other cell types, and cause them to react in a variety of ways. The signal-passing machinery inside the cell can look and work very differently, depending on the intended effect. Here we see a signaling cascade that results in the release of energy molecules. Once liberated, they are sent on their way via the bloodstream to power cells in another area of the body. When this tiny domino effect is triggered inside cells, some amazing things can happen. Other signaling cascades triggered during the stress response result in a variety of physical reactions. This signaling molecule can relax or contract different types of muscle cells. When it reaches the skin, the signal docks on muscle cells attached to a body hair. The tiny muscle is stimulated to contract, raising the hair. The signal also docks on cells wrapped around a sweat gland, which contract too, squeezing sweat onto the skin's surface. In the lungs, the signaling molecule docks on muscle cells wrapped around bronchioles. But these cells relax instead of contract to allow more oxygen into the blood. Because the protein machinery inside each kind of muscle cell is different, this single type of signaling molecule can cause both relaxation and contraction. Many signaling molecules work this way. When this signal reaches the heart, it docks on beating heart cells. Their response is to beat faster. Blood pressure goes up, and the signaling molecule is rushed on to still more cell types around the body. Constantly and in concert, cells must send and receive a myriad of signals. Cell communication is vital to keeping the body alive.
Well, I hope you enjoyed that overview, and I hope to see you later on. So take care and enjoy cell communication.